Bismillahirrahmanirrahim. As requested by some of you, I am doing the specimen paper 2 for the syllabus which is being examined this year in 2022. As you can see, nothing has changed. The timing is the same, 1 hour 15 minutes, and this is the paper 2 for 2022, and this is the specimen paper which I'm going to be discussing today. So timing same, the rest of it is of course the same, 60 marks. As we read the first question, figure 1.1 is an electron micrograph of part of a eukaryotic cell. Of course, this has come in another previous paper, so it's not a new question. And you can see here that they've labeled something mitochondrion and they've labeled something G, and then you can see the magnification here is 47,000 times it has been magnified. Then it says state how it is possible to deduce that figure 1.1 is a transmission electron micrograph and not a scanning electron micrograph. Well, you have to know the differences between them. The scanning electron micrograph is just going to scan the surface of it. So you're going to see the surface features. But in a transmission electron micrograph, you have to see the details of the inside details of it. So you can see the mark scheme is very clear about it. Can see internal structures, or you can say cannot see surface features. Now in another question, I remember you could have talked about this 47,000, because you see this cannot happen in a light microscope. But you see, why can't you put it here in this question is because both are they talking about electron micrographs. So in both electron micrographs, their magnification is enormous. But if you had compared it with a light microscope, then you could have said, okay, well, this is 47,000. In a light microscope, the maximum useful magnification is 1500. So you couldn't have talked about that here. But I don't remember in another question, this was a possible mark scheme. Then coming to the B part of the question, both the Golgi body and the rough endoplasmic reticulum are part of the network of membranes inside cells. Both are phospholipid bilayers. Outline structural features shown in figure 1.1 that identify G as the Golgi body and not the rough endoplasmic reticulum. So let's go back onto G. Now you can see the G part of it here. So this is the G part of it here. Now why is the G part is going to be the Golgi and why is it not the rough endoplasmic? Why? Because they are sacs which have layered appearances. Sacs which have layered appearances, and they are not connected to the outer membrane of the nuclear envelope, or they are the swellings at the end of the sacs, the vesicles at the end of the sacs. We can see those. And another mark scheme point would be that there are no ribosomes on it. So you can see here they are trying to find out whether you actually know what the Golgi body is. And so what are the things which make it sure that it's the Golgi and not the RER? So that was the possible mark schemes for that. Then coming on to C part of the question, calculate the actual diameter XY of the mitochondrion labeled figure 1.1. Write down the formula, you will have to make your calculations, give you answer to the nearest whole nanometer. This is something which you didn't read and you were careless in it. Most of you, if I had given this paper, I know 20% of the class would not have read this. And then when I confront them, they say, oh miss, I didn't read that. So I'm sorry, you cannot say you did not read that. That is a sort of a very, very careless attitude if you say you did not read that. Now, of course, if you give the formula of it, of the actual diameter is equal to image over magnification. Now, everybody should be knowing that. Now, if I measured it and it was 10 millimeter, according to whatever would be on the exam paper. Now, 10 millimeter means first I need to convert it into micrometer. So, I add 3 zeros to that. Then I need to convert it into nanometers. Now, this is where you make the mistake. So, I add another 3 zeros. Where you add the zeros, that's where you keep on making the mistakes. And I know that for sure because I see it every day when I'm checking papers. And then you divide it by the 47,000. Now the answer to that comes something like 0.7, 212.7, 212.7. Now many of you would have written that, but actually it said to the nearest whole. So that would be 213 nanometer. So I mean, all these 2 marks, if you lost those 2 marks, it would be very sad because these are marks which you shouldn't lose at all. Then coming to the D part of the question, many of the cell structures in figure 1.1 are surrounded by membranes. Membranes are approximately 6 nanometer to 7 nanometers wide. So this whole cell membrane is 6 nanometer to 7 nanometers wide. Describe the fluid mosaic model of membrane structures, and there is space below for a diagram. So either you wrote it or you drew it. That was up to you how you did it. But you look at the marks And the marks you can see are 3 marks for this, so please do not lose these 3 marks. Now, as you can look at it, I would have tried to do a diagram so that I could explain these, and I would of course write on it. So the phosphate heads would be all these ones which I am now coloring in black. And then you can see the fatty acid tails are pointing inwards, so I've shown that. I've also labeled them fatty acid tails. And then I would say, okay, proteins, and then the proteins are scattered randomly. So the proteins are, the words used in the mark schemes are proteins present, and then of course you can say proteins scattered in the phospholipid bilayer. So scattered in the phospholipid bilayer, and I would have got my 3 marks. So please be very careful when you are answering these sort of questions. Do you label them? And you know, we call this annotated diagram. Annotated means you've given a little bit of an explanation with it as well, that the fatty acid tails are pointing inwards. But of course that I've drawn in the diagram, but if you had written that as well, I'm sure that would help in whenever you are doing a diagram like that. So 3 marks, and you got your 3 out of 3. Then it says, the part 2 of the question says, the inner membrane of the mitochondrial envelope is much less permeable than the outer membrane. Suggest, again, suggest are the ones which are difficult. So suggest one way in which the structure of the inner membrane of the mitochondrion may differ from the outer membrane to produce a less permeable inner membrane. Difficult question. Have to understand is that why would it be less permeable? So there has to be some reason for that, and that could be there could be reduced gaps between the membrane. There could be more saturated fatty acids, so there's a very closer packing. You see, if you remember, if there are unsaturated fatty acid, means there's a double bond, then there's a kink in the tail. The tail, you know, the normal phospholipid bilayer should be like this, but in this case it will be like this. This is the saturated ones, these ones. These are the unsaturated ones. So that will give you, so more saturated fatty acids, so closer packing, then there may be fewer transport proteins, or there could be more cholesterol molecules. So any of those answers, if you knew the function, only then you could have answered this part of the question. Now coming to question number 2, you can see it's again a question which involves 2 chapters. So the main causes of tuberculosis in humans is the bacterium Mycobacterium tuberculosis. You have to know this name. Most cases of the disease involve the lungs. The bacterium can enter cells. So this bacteria can enter our human cells and remain inactive in a dormant state. In a dormant state means that it's not going to remain, it's not going to be inactive. So you're not going to have the disease, but you will have the bacterium inside your body, in some cells of the body, maybe in the lungs or maybe in other parts of the body as well. However, the bacterium can become active to produce symptoms of the disease. Symptoms of the disease means you'll have the fever and you'll have the loss of weight and all the symptoms which are associated with tuberculosis TB. In a person with active TB, the pathogen can be present in airborne droplets that are exhaled. Like for instance, TB patient is speaking, so these airborne droplets are coming out of his mouth. And if somebody inhales those, then he is going to get TB as well, but maybe not. So it says in a person with active TB, the pathogen can be present in airborne droplets that are exhaled, not exhaled, please remember, inhale, exhale. Generally, a healthy person who inhales these droplets have effective defense mechanisms in the gas exchange system to prevent infection. One example of a defense mechanism against pathogens in the gas exchange system involves the action of macrophages. Describe the mode of action of a macrophage against a bacterial cell. So now this is a long question, and this is for 3 marks. So you know what macrophages do. I'll just quickly revise it and then give you the mark scheme for that. This is a very good diagram. You can see number 1, a bacterium engulfed by a macrophage is encased in a vacuole. Number 2, lysosomes fuse with the vacuole and digest the bacterium. Number 3, antigens from the digested bacteria are presented. You see, if the bacteria is something like this and the antigens are like this, then you can see here the antigens are being presented. So antigens from the digested bacterium. So this is the bacterium and these are the antigens on it, and they are presented, forget the MHC2, on the cell surface. Now this becomes an antigen presenting cell. This becomes an APC, antigen presenting cell, right? Here is another antigen presenting cell diagram, and I would like you to pause the video here and have a look at it. how the antigen, how the bacterium was then engulfed, and then of course the lysosomes digest and all that happens. So just pause the video here and have a look at it. Another diagram, please pause the video here, have a look at it, and go through the different stages. Microbes adhere to phagocyte. Phagocyte forms pseudopores that eventually engulf the particle. Phagocytic vesicle containing antigen. Phagocytic vesicle is fused with lysosome. Phagolysosome. Microbes in fused vesicle is killed, digested by lysosomal enzymes, which the phagolysosomes leaving a residual body. Residual body and the indigestible and residual material is removed by exocytosis. The mark scheme is very clear. Macrophage has receptors for pathogen, engulfs pathogen, forms phagosome, then the fusion of the lysosome, then called phagolysosome. You can of course have said that as well. Then hydrolytic enzymes are released which are present in the lysosome. And then finally this becomes the antigen presenting cell. So the antigens are presented on the surface and forms the antigen presenting cell. Please remember there are about 1, 2, 3, 4, 5, 6 points, but you only have to give me any 3 of these. So not all of them were required. Now coming to the part 2 of the question. Sometimes Mycobacterium tuberculosis survives within a macrophage instead of being destroyed by the macrophage. Suggest one way in which Mycobacterium may survive within a macrophage. I mean, like for instance, if you say, oh, well, somebody shot a bullet, but he didn't die of it. Well, because he must have missed the bullet, he must have ducked or something. So the fact that you have got to give me some suggestions, and this is a suggest question, so it's a difficult question. So maybe it produced inhibitors or it deactivates the hydrolytic enzymes. Maybe they sort of got some inhibitors which are going to make the lysosomal enzymes inactive, or maybe it escapes out of the phagosome. So you've got to think of, very, very weird sort of answers. It's not one answer that I can give you. Or maybe it forms a sort of a resistant spore, means it forms a coating around itself so the lysosomal enzymes cannot digest it. Or maybe there is a malfunction of the macrophage. So any of those questions you've got to think of it is why is it not being destroyed by the macrophage? Then if you know what the macrophage does, then you know what sort of answers you can give, that the enzyme inhibitors for the hydrolytic enzymes or escapes out of the phagosome, or it forms resistant spores. Resistant spores means it forms a sort of a coating around itself by which these enzymes cannot reach it. So suggestions, maybe you could say macrophage does not function. So 1 mark, but be sensible in writing that 1 mark, an answer for that. Then part B of the question, a healthy person has other defense mechanisms in the gas exchange system to prevent bacteria entering cells. Describe these defense mechanisms and explain how bacteria in inhaled air are prevented from entering cells of the gas exchange system. So basically, they were asking you a question about the goblet cell story, which we've all done in our O levels as well. So goblet cells produce mucus. The mucus is sticky, so it traps the pathogens. I don't agree with the word traps because I say, well, the pathogens stick to the mucus, because trap is a very active process, so I don't agree with that. But of course, it's in the mark scheme. Then mucus acts as a barrier because it's going to prevent the pathogens from reaching the cells. Mucus increases the distance to reach the cells because there's a layer of mucus between the cells and the pathogens. Then cilia on the ciliated epithelium are going to move the mucus. You can say move the mucus, or you can say waft the mucus. If you don't know the word waft, then I won't ask you to use that. And where is it going to move it? It's going to move it away from the alveoli and the lungs, and it's going to back to the back of the throat where you're going to swallow it, and it goes into the stomach, and these pathogens will be destroyed by the stomach because you've got the acid and the pepsin in the stomach to destroy it. So basically any 3 out of these, lots of mark scheme points. Then part C, in people with a weakened immune system, Mycobacterium tuberculosis can infect other organs and tissues such as the kidneys and the joints. Yes, I mean, I remember also seeing patients suffering from kidney tuberculosis and bone tuberculosis and even blood tuberculosis. So suggest how the bacteria may spread from the lungs to other organs and tissues. Now that's of course a very simple, of course, where is it going to go travel? It's going to travel through the blood. There's only one way that's going to travel. You could have said the blood, you could have said the plasma, you could have said the circulatory system, or you could have said the lymph. So both were correct, or you could have said macrophages will travel or phagocytes. So I mean, but the blood and the lymph were the two most common ones. Then coming to the D part, TB in humans can be caused by another species of bacteria, M. bovis. State the mode of transmission of this pathogen to humans. Now, basically, you know that is eating contaminated meat, or you can say eating meat that is from infected cattle, or you say drinking milk from infected cows. So that was an easy question. I don't think that was a very difficult question. Eating contaminated meat, or is saying eating beef or meat from infected cattle, and the other was drinking milk from infected cows. So that was the answer to that. The question, it says rifampicin is one antibiotic that can be used to kill mycobacteria cells. Although rifampicin is very effective at killing mycobacterial cells, it is often the first antibiotic to which resistance develops. Now, again, the question syllabus is antibiotic resistance. Rifampicin binds to a section of RNA polymerase that has attached to the DNA template strand. Now, remember bacteria, the DNA, and so there would be this gene for some RNA polymerase. So now what happens is it binds to a section of RNA polymerase. So this would have been created for RNA polymerase, would be the enzyme present in it. So it attaches to the DNA template strand. Explain how binding to RNA polymerase allows rifampicin to kill the mycobacterial cells. Now, of course, we need to revise this. Now let's try to understand this. Now I've drawn this. This is a bacteria, and this is the bacterial DNA. Now, when any protein, say this is the portion which is being transcribed. Now, when what we need is from this part of the DNA, we need mRNA to be made. But for this mRNA to be made, you need an enzyme called RNA polymerase. You need an enzyme called RNA polymerase. So from the DNA, this is a gene which is going to make an enzyme or a protein which the bacteria needs. So this is the portion of the DNA which is being copied by the mRNA. So this is the mRNA being made. And we need the enzyme RNA polymerase to do this. Now it says rifampicin binds to a section of RNA polymerase. So the rifampicin binds to something like this is rifampicin, so it binds here. Now explain how binding to RNA polymerase allows rifampicin to kill the bacterial cell. Why would the bacteria be destroyed? Why? Because no mRNA would be made. No mRNA would be made. So no proteins would be made. And the bacteria needs proteins for its growth. It requires the enzymes for growth, for replication, for its own metabolism. So no proteins will be made because the RNA polymerase is not functioning. It's just like if you didn't have pepsin in your stomach, well, then how would food be digested? So if somehow pepsin is disabled in our stomach, then we won't be able to digest any protein, so we won't get amino acids. The amino acids we need for growth, to make new enzymes in our body. So basically, this is what they were asking you in the question. So mRNA is not synthesized, and proteins or enzymes required for growth and replication will not be made. Then going on to the next part of the question, the standard treatment for TB continues for 6 months. Initially, 4 antibiotics are prescribed. This is then reduced to 2 of the 4, rifampicin and isoniazid, if the person responds to treatment. A person with MDR-TB, which is called multi-drug resistant TB, does not respond to treatment with rifampicin and isoniazid. The treatment for MDR-TB involves other antibiotics and lasts for up to 30 months, 2.5 years. Table 2 shows the number of reported cases of TB and MDR-TB in the Southeast Asia region between 2005 and 2014, as published by World Health Organization. Now, as you look at this table, table 2.1, 2005, total number of reported cases was 19,47603. Total number of reported cases of MDR-TB were only 68. In 2006, 21,04673, so it has increased, but these have also increased. Then 2007, this has increased a little, and then this has increased to 918. 2008, 2287, 1717. 2328, 230, 2560. Then 2332, and then 4263. Then 2358, 127, and 6615, 6615. Then 2331. Now here you see something weird is happening. and 14,957, 2297, 033 and 18,384, and then 2580605 and 17,000. Now you can see some sort of a trend and look at it carefully, read it again and mark things. Now the question is table 2.1 shows that between 2005 and 2014 there's an overall increase in the total number of reported cases of TB and MDR-TB. Describe the differences between the trend in the total number of reported cases for TB and the trend of MDR-TB as shown in table 2.1. So let's go back to now they've said, okay, then overall increase, but differences, now this is where you have to pick up the differences. Now this was very simple, wasn't very difficult. Let's go back to the table. Now what were the differences? Now the rate of increase in cases of TB greater than rate of increase of MDR-TB was, look at the MDR-TB. So the rate of increase in MDR-TB is greater than the increase of the normal TB. Rate of increase, this was 68 and now it is 17,000. This was 1947603 and now it's 2580. So the rate of increase in MDR was much more. So this is what you had to pick up from the table. Which was there was a decrease for TB between 2011. There was a decrease. So you have to see is, you see now if you see there is a decrease of TB between 2011 and 2013. TB. But the MDR-TB shows a decrease between 2013 and 2014. I mean, of course, many of you can say, Miss, this is no biology, it's just looking at a graph and interpreting it. Yes, well, that's part of the syllabus, interpretation of graphs. So, decrease of TB was between 2011 and 2013 here, this point. And MDR-TB was between 2013 and 2014, which you could have said also decrease over 2 years for TB, whereas only 1 year for MDR-TB. And then, of course, you could have said another thing, which was also possible, is that it shows a plateau. Reported cases of TB show a plateau between 2009 and 2012. You see this area. It's not very increasing a lot or decreasing. So it's a plateau between 2009 and 12. But in MDR, there was no plateau. There's no such thing seen. So you could, this is of course a very, very important skill of biology which they are testing, which they have put in the syllabus, that you must be able to interpret and be looking at tables and find out differences, etc. The last part of the question G, many social and economic factors need to be considered in the prevention and control of MDR-TB. Outline 3 of these factors. So that was a very simple 3 marks for this part of the question. They're asking you, number 1, economic, social factors in the prevention and control. So 3 of these factors is, number 1, you could have said DOTS. The DOT strategy for normal TB will reduce the risk of MDR-TB. DOTS was what? Direct observation treatment short course, in which you ensured that the patients took their medicine. Basically, why is antibiotic resistance increasing? Why? Because MDR-TB is increasing because people take the medicine, get better, and then leave it. They don't take it for the prescribed time, like 6 months to a year. So this was the problem. So if we prevent this problem in the beginning only, then we can prevent MDR-TB. So number 1, DOT strategy. Number 2, improving the accommodation so that people who live together in small places then spread the disease. So we would improve that to prevent the spread. Then education of the people to prevent the spread. People, sufferers, should be educated that they should not be spreading it to their relatives or whatever. They should sort of abstain from meeting people. So education of the people to prevent the spread. Then particular social groups like HIV infected people are at a high risk of tuberculosis. Then if we target those people, and then we use less antibiotics in agriculture, so that's another point. But then we think of the cost, because they've also asked you economic. They've also asked you here, they've asked you economic factors. So economic factors, then you've got to think of the cost. Now, when you think of the cost, vaccination programs are going to cost a lot of money. Then if we give people longer drug treatments, that's going to cost a lot of money. Then if we research into new antibiotics, that costs a lot of money, and the money is diverted from other health issues. Then educating the healthcare personnel and the veterinary people that, okay, if there is tuberculosis in the animals, if there's tuberculosis in the cattle, then you need to actually not sell the meat of that cattle and not sell the milk of that cow. So this is a long term. So this was a many questions. There were many possible answers, but only the student who understood the question, that social and economic factors to be considered in the prevention and spread and control of MDR-TB. You see, when was MDR-TB going to affect you? MDR-TB was going to affect you when people had become resistant to TB, or the ones who were suffering from MDR-TB then spread it to other people. So unless you understood the question, you would not be able to do this. This was a very long question and it had 16 marks. So this was really checking basically the two chapters, which are the last two chapters, diseases and immunity. I will conclude this video here and I'll continue with the rest of the questions in another video which will precede this. So all the best and thank you once again.